Good morning, everyone. Thanks again for joining us, for being here today. We are very excited to have Dr. Wang joining us today for our Distinguished Speaker Series. I'm Nancy Walters. I'm the Executive Director of the La Jolla Community Center. And if you are new to the Community Center, welcome. We are a nonprofit organization and with the mission of providing programs and services to promote lifelong learning, wellness, and friendship. And we host a variety of programs and classes ranging from language and fitness and wellness and series. We have author talks, concerts, and more. Most of our daily programs are back in person. We were closed for about 3 weeks, but we are now back in person. And we're also hosting some classes virtually, just like this series. But please visit our website where you can see our full schedule of current events and programs happening here at the center. Our website is ljcommunitycenter.org. I hope that you can visit us there soon. Before I introduce our speaker, Dr. Wang, I want to remind you that you have the ability to send in questions for the speaker today using the Q&A function that should be at the bottom of your screen somewhere, depending on what device you're on. So please feel free to use that Q&A function on your screen, and we'll get to as many questions at the end of the presentation as we can. So it is my pleasure to introduce Dr. Joseph Wang. Dr. Wang is a distinguished professor of nanoengineering at University of California, San Diego. Dr. Wang earned a doctorate in science from the Israel Institute of Technology. He has served as a research associate at the University of Wisconsin and was a member of the chemistry department at New Mexico State University, where he held a Regents Professor and a Manessy Chair. Dr. Wang also served as a director of the Center of Bioelectronics and Biosensors and a professor of chemical engineering and chemistry at Arizona State University. He joined UCSD Jacobs School of Engineering in 2008 and served as the chair of the nanoengineering department between 2014 to 2019. Dr. Wang has been the most cited scientist in engineering in the world since 1991 and first on the ISI chemistry citations list. He is the author of over 1,200 papers, 10 books, and 30 book chapters. Wang is the recipient of many international awards, including a 2007 National Science Foundation Special Creativity Award, the 2006 American Chemical Society Award for Electrochemistry, the 1999 American Chemical Society Award for Analytical Instrumentation, and the 2019 Outstanding Sensor Award of the Electrochemical Society. Wang's research interests focus on electrochemical biosensors, wearable devices, and microbots. So please help me welcome Dr. Joseph Wang.
Thank you all for coming this beautiful day. Instead of being on the beach, you come to my Zoom talk. So again, I'm a professor in UCSD. This is our famous library. And let me check. And we are here in La Jolla, and why UCSD? Why we are doing all this digital health in UCSD? Now, we are International Center for Biotech and Wireless. We have Qualcomm, we have Illumina, we have Dexcom, and all on the Mesa in Torrey Pine, we have the Salk Institute, the Scripps, all surrounding our beautiful campus. And UCSD in our top ranking, we are a young university, but we already number 4 in the ranking in the best public university. Our bioengineering department is number 2, and I'm excited to be the director of the Center on Wearable Sensors, CWS. I have 20 faculty in my center, and we are striking to meet the challenges of developing next generation of wearable sensor. We have, again, a faculty for 6 different departments, including our dean, people for electrical engineer, bioengineer, medicine, design, all working on cutting edge research in the field. Now, this field is exciting. It's been now almost 2 decades of digital medicine, all these gadgets, the Apple Watch, etc. And this is now aiming to go to personal wellness and personal medicine, because we are all different. Every person is unique and different. So one treatment will not fit all of us. Because of the uniqueness on personal medicine, we have to have different personalized tailored treatment. We have to do the diagnostics so we can tailor the therapeutic for individual patient. And this wearable electronic is expected to measure a key role in this move to personalized medicine, because we are now measuring continuously many vital signs and many chemicals during daily activity non-invasively. So again, this started about 15 years ago, all the digital revolution with many gadgets that you see here. But the early stage was mainly to looking at steps and vital sign, ECG. But this device can monitor continuously your daily activity and have the potential to improve the quality of our life while reducing the cost. So again, the early work was focused on vital sign. And again, we still measure our steps continuously, but what was missing is the molecular information about molecule, like glucose. Glucose, we see a lot of interest in diabetic, but there are many other molecules like cortisol for stress, or lactate for performance, of sodium for hydration. We need to capture this molecular information continuously. So the question, my lab, we pioneered this field 10 years ago, to get molecular sensing, and the goal is to digitize the molecular world. We want to have continuous 24/7 monitoring toward this personalized medicine, personalized nutrition and wellness. So we want to go beyond the mobility and looking at molecular information, dynamically changing biomarker. So we have this device that monitor simultaneously vital sign. And this is from the cover of Time magazine 2014, where they show that this in the future will be never go offline, all day monitoring. So this was nice cover, and they say how the wearable technology will change our lives. So this was almost 8 years ago, and we made a lot of progress on this skin device, lab on the skin. So what we have currently, we have devices that monitor our motion, the steps, ECG, maybe skin temperature, respiration, but even blood pressure. These are all physical vital signs. What is missing is the molecular information about biochemical, such as electrolytes, sodium, potassium, stress, cortisol, alcohol, and obviously glucose for diabetic, and many, many more. So this is a great opportunity. It's still missing, even on the Apple Watch. We still don't have a glucose sensor. We have a company like Dexcom, they have a big needle. They are not wearable in terms, they are very invasive because they are 10 millimeter. We want to make it minimally invasive or non-invasive. So this is a great opportunity to include this molecular information and combine it with the vital sign, physical and electrical attribute. And this is what still is missing, the lack of information about chemical constituent like this one. So it's indeed great opportunity, and it also was accelerated recently by the pandemic. So all the move into telemedicine, remote medicine, has really been a major driving force over the past 2 years to push this field even more and more into telemedicine and remote medicine that combined with wearable, people will monitor their body continuously, and this is remotely sent to the nurse or caregiver or the doctor. And this is now pushing the field of remote healthcare for the future. And the vision is, if you go to the standard laboratory, there are big benches with a lot of equipment. And our vision is in 10 years to shrink all of this onto the skin or even into the mouse, to have a complete laboratory instead in the centralized lab, to shrink it into the mouse, on the mouse guard or in the arm, and use it to check your engine, like you check your car engine. You'll have a warning and prediction of any disorder. So the goal of this lab on the body is to really not only to monitor problem, but in advance to predict problems so you can take intervention and give you the check the engine light to take a precaution. So this is the vision of shrinking the lab onto our skin or onto our mouse. And this has many advantages, giving you the chemical information continuously without taking any blood sample. And again, it will be much cheaper, remotely convenient, better compliance. So we will collect very rich molecular information that at the moment you need to go to the lab and take your blood sample. And you can do it continuously during daily activity. So these have also some challenges. These are not straightforward. They have challenges of stability of the sensor. You need to validate it compared to the gold standard blood. You have to have issue if you put the sensor in the mouth or on the eyes, data security, safety, movement of the body. Sensor have to be flexible and stretchable. So all of this have to meet the accuracy of standard lab. You cannot compromise the performance by doing on the skin. So many challenges you can see here, and we are tackling these challenges. We are using electrochemical device. This is like the Dexcam device from San Diego for glucose. It's also electrochemical, but this is a large needle. We are trying to make it on the skin or on the textile, because these techniques are portable, have low power requirement, and easy to use. Now, this field of biosensor is already 60 years old. From the first glucose sensor was introduced in 1962. And it is a long time from moving from the lab bench to the body. So during the 70s, we saw a bench top device. The 90s, we see this disposable strip DNA sensor, but only the last 10 years, we moved to this wearable electrochemical sensor for biomedical information. This is in my review in Nature Biotech. So we are moving from the lab to the body. And if you visit my lab, or you can visit me in UCSD very soon, you'll see my different mannequin with different prototype, sensor on different accessory, on glasses, on textile, on glove, on ring, a variety of sensors for biomedical, wellness, nutrition, and even security application. And you can see a sensor on socks because we have a lot of sweat, a sensor in the mouth, and so on. So basically, we are trying to have non-invasive analysis of different body fluid. Instead of blood, we will try to do it on sweat, like this beautiful flower tattoo sensor, saliva, this is this mouth guard. We can do contact lens sensor for tears analysis. We can do microneedle sensor for interstitial fluid under the skin. And we can also monitor the surrounding environment for explosive or for nerve agents. So in all cases, we are analyzing sweat, saliva, and tear, looking for different molecular biomarkers for disease, nutrition, and wellness. So the type of analyte we are looking at a wide range, again, metabolite like glucose, lactate, alcohol, electrolytes, sodium, potassium, a drug like levodopa for Parkinson, or a drug of abuse like opioid, a stress marker like cortisol, hormone like testosterone, different vitamin for nutrient, again, substance of abuse, chemical agent, toxic metal, explosive, and so on. So by doing so, we can get information about our health, wellness, performance, and security. So the technique that we are using to put sensor on textile is printing. We are using screen printing. It need to be flexible sensing electrode, sensing array. Here we have example of 8 electrode on textile for military application, on Gore-Tex textile. So we are transferring this pattern of electrode. You see this, these are 8 carbon electrodes. We transfer it to the textile. These are the silver contact and so on. We're putting the ink, and we are pressing it on the textile, like screen printed of t-shirt. And we can do it on skin, we can do it on tattoo. And the challenge, these electrodes usually are very rigid. All these devices are very rigid. The problem that the biology is very soft. If you visit the San Diego Zoo, you see this flamingo near the gate. They are very stretchable, very soft and very stretch. You see the body, the babies are all soft, but the device usually are very rigid and planar. Our goal is to bridge the gap between the softness of biology and the hardness and rigidity of this device. So we are developing flexible device and can bridge the gap between the two and be compliant with the body. So for this, we are using innovation in material. We are developing device on textile, on skin, device that can stretch, can bend, can fall, even self-repair. So we are bridging the gap by using this stress-enduring material that can indeed stretch and twist and bend. Here you see how we're doing it. We're printing this design, this serpentine design. This serpentine can stretch when you put it on the skin, when you twist it. So you're pushing the ink on the stencil. And by doing so, you can see we can stretch it 100, 200%, push it really to the limit to have a lot of resiliency for mechanical stretch. And this keeping the electricity, the conductivity, even if you push it to extreme stress. So this allows us to go to put our device on the skin, because the skin is the gateway to our body, to our biochemistry. Using the skin, we can analyze the sweat, or we can analyze the ISF, interstitial fluid under the skin. So we are developing non-invasive devices like tattoo, which are flexible and can analyze the sweat or ISF. And eventually we have a complete lab on the skin in 10 years, with all the electronic connection, the power, wireless communication, not only the sensor, but our whole skin will be a laboratory with the powering, flexible battery, or it will be electronic textile. All our fabric, the clothing will be look like this. So to do it on the skin, like this is a nice smiley pH sensor, or this is a flower shape, a lactate sensor. We will go to La Jolla Shore. In La Jolla Shore, you can buy this tattoo. And we are getting this tattoo, which are very elastic, and we are printing our sensor in a nice smiley shape or flower shape on this tattoo. And this combines the tattoo technology with our printing technology. So you can see this, you need the 3 electrodes in this flower. We have 2 carbon electrodes and silver. You transfer it to the arm, or this is the smiley pH sensor. We have 2 electrodes, the 2 eyes. Using this technology, you see this is a glucose tattoo printed on the paper. And then we will transfer it, you'll see in the video, to the skin. So this is low cost, it costs a dime to print it on this, and then you transfer it to the skin, you peel the protective layer. And this is in slow motion, usually it takes 5 seconds. And we will use it to monitor your sweat glucose non-invasively. So this is the tattoo for glucose sensing. It also can extract the ISF. We will put the electronic here. It's very low cost, and you can use it all day to monitor glucose non-invasively every 20 minutes. So the way you put it on the skin, then you put your electronic. The electronic is reusable. The tattoo is used for 1 day. There is magnetic connection, so you can place the electronic directly. These are flexible electronic you see here. So here we extract the fluid from the under the skin to the electrode with the enzyme. This was displayed already in Vienna, in the Technical Museum in Vienna. You see the diabetic plaster. They show it future of medicine. So by doing so, we can, again, moving to a glucose from the traditional bench. This was in the 70s, was testing in the bench. This is the 90s where we do the strip. Now we are moving to Dexcom, which is on the body. But again, this is still invasive, 10 millimeter long. Our goal to make it non-invasive, like we do it here. So we will get the glucose data on your smartphone without even piercing the skin. So this is, we'll go to lunch in 2 hours, and then you can put my tattoo, and you can, in 2 minutes, you can see the change of the signal before the lunch and after the lunch. So basically you have this all-day tattoo measure every 20 minutes. Again, the electronic, the power, wireless communication. You have an enzyme, it specifically recognizes the glucose. So the glucose oxidase give you specificity. And then you see the increase in the signal after this big lunch. Also, if you go after dinner, you'll have your drink also. You can also measure your alcohol, your sweat alcohol with a similar tattoo, but instead of enzyme for glucose, now we have enzyme for alcohol oxidase. And here you stimulate the sweat, and in 1 minute you get the sweat on the electrode. And then you have a reaction, and in 2 minutes, it will tell you how drunk you are. So overall, you stimulate the sweat and measure, it take 2 minutes, and then the telephone will tell you you are drunk, don't drive. And you see the correlation to standard technique. If you are not drinking, there is no signal. When you drink too much, you have larger signal, drinking less. So this is our glucose sensor, similar electronic, but different enzyme. Then we combine the two in this nice panda shape. We have a glucose. What we do, glucose, we measure the sweat alcohol and ISF glucose. One electrode is for the alcohol, another one is for glucose. So this panda can extract both the sweat and the ISF and measure on this eye, we measure the glucose. Here we measure the alcohol. We have different enzyme. You see, you can twist it. Here is the electronic, and then you eat and drink, and you see the change of the signal of alcohol and glucose. This again, fully non-invasive, and you can twist it, you can stretch it, and you get the readout in 5 minutes, both the glucose and alcohol level. Now, a lot of interest in personalized nutrition, especially vitamin C and D. With the pandemic, they support the immune system. We work with major company in Switzerland, DSS, to analyze this vitamin in sweat, non-invasive. So here we have a tattoo. You can see it here for ascorbic acid, which is vitamin C. And you can follow the kinetic, the uptake of the vitamin by the body by analyzing the sweat. So this is again electrochemical detection. You have specific enzyme, and you do measurement every 20 minutes after you take the pill. You take standard pill, but different people respond differently. You can see the profile are different because of this personalized nutrition. You want to tailor the nutrition to the person based upon their specific kinetic metabolism of the vitamin, and to use it to guide your diet. So this is the major goal in our lab for personalized nutrition, not only for vitamin C, for all. You combine it with picture, combine it with computation like machine learning, and analyze these multiple vitamin and electrolytes, sodium, potassium, glucose on multiple sensors on the skin using this array, and taking all this information and do what we call data fusion and data mining with machine learning. And this will guide you your diet toward optimal nutrition on the personal level. So we are continue personal nutrition, personal wellness. We also combining this chemical sensing with the vital sign. So remember, the early day we did the ECG or EEG or blood pressure without the chemical. Now we are developing tattoo. So on the same tattoo, we combine the chemistry like lactate with the heart rate and ECG monitoring simultaneously. You see the electronic and these electrodes are the ECG electrode. In the middle are the electrochemical for the glucose or lactate. And there is no crosstalk, and we can do it simultaneously. So we are fusing the vital and the chemical sensing. More recently, we published this paper in Nature when we combine blood pressure monitoring with chemical sensing. So this is what we call acoustic blood pressure tattoo. Combined with chemical sensing of ISF or sweat. So you see it sending signal, monitor the blood vessel contraction for the blood pressure. At the same time, we can measure ISF, glucose, or sweat, alcohol. So you can monitor daily activity. And by doing so, you have simultaneous monitoring of blood pressure and multiple biomarkers. You take your coffee, you can measure caffeine or stress. You have your cheesecake, you can monitor your glucose, your exercise. You see the blood pressure is changing, also the lactate is changing. So this allows you to have more comprehensive monitoring, all in a flexible tattoo that, again, these are the chemical sensors, these are the acoustic blood pressure, and you can follow your daily activity. If you're obese, you can see how the blood pressure, you have cardiac problem, also diabetic. So both looking at sweat biomarker, ISF biomarker, and the blood pressure changing. So you see how it looks. It's about 1 inch wide, 1 inch square. These are the acoustic blood pressure sensor. This is the ISF glucose. This is the sweat lactate, very flexible. And by doing so, this is example when you exercise. When you exercise, the sweat lactate is increasing. This is beginning of the exercise. You get the sweat, the lactate is increasing. Then you stop exercise, it's falling down. At the same time, the blood pressure is also increasing. Before the exercise, 75, and then go to 110, and then going back. And this is all validated with standard lactate or standard blood pressure sensor. So this is the latest innovation. We're also trying to, instead of exercise, to get the sweat from the fingertip. Fingertips, there is a lot of, actually the most sweaty part of your body. The largest sweat rate is in the fingertip. And here you don't need to exercise. You don't need to stimulate the sweat. You can just touch the sensor and you get your sweat glucose or sweat alcohol or sweat dopa, as you'll see soon. So this is a touch-based sensor that you don't need any activity to stimulate the sweat. Here is an example of glucose. We touch the sensor like this. This is portable device, and there is an algorithm that personalized, correct for any skin variation. So it translates the readout of the glucose to the blood. One-time calibration, the beginning of the week, you can calibrate and then use it, repeated touching without any blood the whole week. So this is basically eliminate the need for blood-free diabetic testing, no pain, no blood freaking. So just simple touch, you get rid of all of this, and you analyze frequently the glucose with good correlation to blood glucose level. We can do the same to do stress monitoring by monitoring your cortisol in sweat. This is what we call a stressless stress testing. So you don't need any stress, you just relax, put your finger on the sensor. The sensor is specific to cortisol, and this is very low concentration, and you can do it at the home with a small portable instrument. Or we are working with the Parkinson department in UCSD. We are developing similar touch-based sensor for levodopa, the drug. So you take the pill of the levodopa pill, and you see in 40 minutes it going up and down. Just repeated touching every 10 minutes, you see the sweat profile is increasing, and then it's all already degraded. But the initial profile is very important for tailor the treatment of Parkinson patients. So we are working on this, and this is correlated to the blood, but this requires no blood. So this is one activity. We also use accessories like ring and glasses. So here we put, we go to UTC, we buy some fancy glasses, we put the electronic on the arm. We have two sensors, one for electrolyte and one is for metabolites. So we have different electronics for the electrolyte, like potassium. Here is for lactate on this side. And then you put the sensor here, as you can see in this video. You place the sensor on the tip of, like this. This is the glucose sensor, then another sensor for potassium. And you monitor simultaneously the potassium and glucose using these glasses. As I mentioned, electrolytes are very important for many disorders like kidney disease, dehydration, predicting a fall prediction for elderly. So monitoring electrolytes, sodium and potassium, is extremely important. This technique is what we call a potentiometric, ion selective electrode. Now, traditionally, these electrodes are very rigid, made of glass. We convert them to our tattoo. As you can see here, we have our smiley pH tattoo, measure the pH. So we go from the rigid glass electrode to a flexible tattoo. And we put it on the skin. We measure the pH profile before you do many twisting of the skin. So you can see it smiling in the beginning, smiley after 50 bending of the skin. This is the bending. 50 bending, repeated bending, it's still smiling like this, and it's still responding. You see, they respond before and after this repeated bending is still the same. So we have the elasticity of the tattoo. We also do it on textile. This is electrolyte, sodium and potassium electrode with a combined reference on different textile. You can do it on the underwear, where you have contact with the skin, on different wristband, again, sodium, potassium sensor, even in your socks. So this is all fabricated on a flexible textile. We can also make a fluidic for sodium and potassium with the two electrodes. The sweat is pushing over we have a dye to demonstrate. So the sweat is pushing over the sensor, monitor your sodium and potassium, and this is replenish the sweat, so you have continuous sweat flow over the sensor. Again, one for sodium, one for potassium. And you can do the same for electrolytes. So this fluidic approach, we call epidermal fluidic. Here we put enzyme for lactate or glucose. And again, we have 4 inlets for the sweat, it's pushing the sweat, and you have the enzyme for lactate. It can measure once your sweat arrives, you see the profile of the lactate. And this technology from UCSD is recently licensed by a Swedish company, Innosense. They already have their, Innovosense, Innovosense. They have a very nice Swedish design called SMASH, for sweat metabolite else, and so this was just introduced, their product in Sweden. We're also using this technology for monitoring wound healing on a bandage. So we print our sensor, our pH sensor, on a bandage to monitor the wound healing. pH and uric acid are very important biomarkers for the wound condition. So you can monitor the wound condition continuously by putting the bandage. Here we have uric acid. There's another important biomarker. We have enzyme called uricase. So these are the key biomarkers, and you can follow the progress and the healing of the wound by continuously monitoring. And you see, these are very flexible bandage. Here you see the pH after 100 bending, every 20 bending, the same respond. We also developed this bandage for screening for skin cancer, for melanoma. So when you go to the annual, the skin doctor will look at the mole. But here we provide molecular information because in this melanoma mole, there is an enzyme biomarker called tyrosinase. So by placing the bandage and you have a product, we have a reaction specific for this enzyme. Only in the presence of this melanoma biomarker, you get this generation of this product using this enzyme. And this will see increase in the signal, which tell you this is suspicious melanoma. So in this case, it's a quick testing, take 1 or 2 minutes. It's just preliminaries, and the doctor will have a biopsy to confirm it, but at least it gives you rapid screening of potential skin cancer using this bandage approach. We're also working a lot on sensor in the mouse, putting all the electronic on this mouse guard, or even tattoo on a single teeth. So there is a lot of salivary biomarkers. Saliva is very rich with different biomarkers like cortisol, like lactate. So we can have all the electronic, all biocompatible and safe, either on a mouse guard form or even this tattoo form. We and other group, especially Google, started with this contact lens, and actually they stop it because they have some issue. But the contact lenses are very attractive for tears analysis. So they are sensor developed not only by Google, a group in UW. We are working here as a Swedish group working on analyzing tears using different enzymes in this contact lens form factor. We also have security application using our gloves. We have 10 fingers. Every finger can analyze a different agent like nerve agent, explosive agent, drug like opioid. This is work with the DOD or DITRA or the forensic agencies that you swipe the surface for gunshot residue or looking for a pesticide or nerve agent residue. I have 1 finger to do the collection and 1 the detection. And here is example of this nerve agent glove. You see, we print it with this serpentine. Serpentine structure allow you to stretch. The electronic is on the ring. So 1 finger is collecting the, let's say, the pesticide from the tomato. Then you close the 2 fingers and bring it to the enzyme, and then you get a signal if there is a pesticide like Parkinson. But we also use it for military application for detecting nerve agent threats. So the Army is very much interested in this to protecting our soldiers and so on. Again, so we're building a complete lab on the skin. We can also have the lab under the skin. Under the skin is even more information. Again, you need to convert all this lab under the skin in this 1-inch diameter. And to do so, we are developing integrated microneedle. Microneedle are an array of multiple electrodes, sensing electrode. I can have on this 1 inch, I can put a 64 electrode for multiple analyte, glucose, lactate, alcohol, potassium. So this is approach to create lab under the skin. They are minimally invasive. They are painless. It's less than 1 millimeter, so you don't feel it, and you introduce it to the skin in a painless manner. And again, these are completely painless and multiplex. You can measure multiple. Here we have 12 sensors, each of them individually addressable. So you're just packing different reagent, different receptor to get this selectivity for different analyte. So you can monitor simultaneously, painlessly, minimally invasive, multiple, let's say, diabetic biomarker. We have a major project with the Emsley Foundation for diabetic management. And this is not only monitor glucose, but other biomarker like ketone. ketoacidosis is very important complication of diabetes, so we measure the ketone. It's also important for wellness and nutrition, but for diabetic, we do glucose, lactate, alcohol, and ketone. And again, this is a 12 millimeter diameter, 1 millimeter height. And here we have example of a dual analyte glucose ketone needle. So we have the two sensor, one is for the ketone, which is the biomarker, hydroxybutyrate, and one is glucose. We have different enzymes, one for glucose oxidase, one is HB dehydrogenase. And by doing so, we measure simultaneously the ketone and the glucose without any crosstalk. These sensors are individual, they're independent, there are no cross-reactivities. So this is the ketone sensor. You have alternate addition in each of them responded, and we are expanding it to more analyte like insulin and cortisol and so on. We're also working on microneedles for continual level DOPA monitoring. In this case, again, monitoring the Parkinson drug level DOPA continuously compared to the strip before. Now we can have continuous measurement every 1 minute interval. You see the signal of the level DOPA. We have two different detection schemes on the same microneedle. One is direct detection, one is indirect, enzymatic. You get more information. And the goal of this is to have a continuous monitoring of not only DOPA, other drugs like apomorphine related to Parkinson, and combine it also with other physical sensors for Parkinson, movement sensors, a variety of other sensors, and taking all of these sensor network and develop a closed loop system similar to diabetic, similar to artificial pancreas. Here we are developing a closed loop that take multiple sensor, level DOPA, movement sensor, get all this information, fuse it and process it in the cloud, and then making decision for tailoring the delivery of the drug in response to this sensor. So it's a closed loop, similar what we do for glucose in artificial pancreas. In glucose, we deliver insulin. Here we correct we have a level DOPA pump to correct it in a closed loop fashion. So this microneedle are, if we are fully integrating it, not only the sensor, which is shown here, we are putting the electronic, we put the battery, this is my nano bioelectronic lab. So all of this is fully integrated with wireless communication. And this led to several spin-off companies from my lab. This is one of the company, many of you heard maybe on BioLink. From my former postdocs, they're now doing very good. They soon will be launching their glucose sensor. They recently got over $100 million 2 months ago from investor to push their first microneedle glucose sensor. So they are up in near UTC in La Jolla. And we have two other companies, one of them in Australia that focusing on nutrition called Nutromics. All of these sensors, as I mentioned, not only require good sensor, but also you need the energy, you need the data security, wireless transmission. So we've been doing a lot in this field, especially I'll give you in 5 minutes few examples of energy, meeting the energy demand. We try to power the device, not using battery, but maybe using energy from our body. We are putting a tattoo-based biofuel cell that you can harvest energy from sweat lactate. There are a lot of lactate in sweat, and you can use it to power the device using biofuel cell tattoo or biofuel cell textile. You can see this large biofuel cell. You're shown here, it's very stretchable. You see this serpentine with two anode and cathode, and then you can power it. You get your sweat, you can power your LED. You can see how the sweat is powering the LED during the exercise. So we're just using the sweat energy. There is no battery to power different devices. And we can combine this energy harvesting. We also can do it from sweat fingertip. Remember, here you don't need to exercise. You can even go to sleep. We call it dreaming power or power of dreaming. You can get the sweat continuously while you're sleeping for 8 hours. And this is what we call a large return of investment. You don't need to exercise. You get your energy without any investment, without any stimulation. So this got a lot of attention recently. It was in the cover of Nature magazine. Say, while you sleep, device can harvest the science. Talk about your sweaty finger can power next generation electronics. So again, we are using this natural sweating to power our electronic display and so on. And we combine this biofuel cell with energy storage, like a supercapacitor. On the same textile, we have one side is the biofuel cell and the other side is the supercapacitor. So whatever energy you harvest from the sweat, you can store it for future use using this device. You can see it's also flexible on the fabric, very stretchable. You can twist it. This is the biofuel cell. You can see the other side is the supercapacitor. So again, it's a hybrid energy system, highly stretchable, getting the sweat energy, and this is the biofuel cell side, and you store it on the opposite. You see the serpentine structure to allow the stretching and bending. So this just example, we are combining all of this, we developing a complete microgrid, like a city grid. We have a smart grid on the body to power this device anytime. This is another Nature paper where we show we can combine multiple energy sources and multiple energy storage on a textile, and you get your power anytime from sweat or from movement, and you can store it like in a grid system, on-demand power supply. So I think my 50 minutes are getting to an end. So I hope that I gave you a flavor how we are bridging the digital age with molecular information toward developing remote personalized medicine, personalized nutrition and wellness. Basically, we have continuous non-invasive chemical monitoring for remote medicine. And eventually this wearable sensor will digitize the molecular world, and again may give you an early warning and prediction before any symptom occurs. So the goal is to get a trend, looking for a temporal trend to multiple biomarkers that allow you to predict and give you warning of potential symptom, or even like COVID-19 and so on. So this is my message. This is the people that did the job before the pandemic. We have 40 people from 20 different countries with multidisciplinary. They did the job. We have excellent collaborator, support from different government agency, different company, again, the people, and my center, Institute of Medicine. And thank you all for your attention. Thank you very much.
Okay. Thank you, Dr. Wang. That was fascinating. Thanks for sharing with us. We have a few questions. There's one that says, where can one see these devices? Are they in your center, or are there any real-time demonstrations for the public?
Yeah, the same. We can arrange, if we have some VIP visit, we have some mannequin and you can touch and feel it, so we can arrange. Now it's little with the pandemic, a little more, it used to be more open until the pandemic, so we need to coordinate it. At the same time, a lot of this was on my website, and also I mentioned some spin-off companies already, hopefully we'll see it in the market. So we are getting closer, but we'll be happy to arrange an individual visit considering the safety with the pandemic. So please contact me to Joseph Wang at ucsd.edu, yeah. So this is Glenn. Glenn have a question. How do you respond to the fear of people? Yeah, this is a good question in time. And there is a two way. One way, you don't want the you want to keep it very safe in general. You don't want artificial pancreas that somebody can sabotage and affect your insulin delivery. So you want to protect it in general. But I know in the case of sport, we have in touch with the NBA team. Many of the players don't want this information will be shared. So there are some hesitation, the same like vaccine and so on. But again, a lot of this in the sport and wellness, they're moving a lot, the personal nutrition. So this will be all protected like any other wireless information. So people are moving to, as we mentioned, personal nutrition, personal wellness, sport and fitness. But again, always the risk of safety, I agree.
Okay, additional question. How do I access your Parkinson's dopamine monitor at UCSD?
Indeed, we are closely work with Dr. Irene Litvan in the Center of Parkinson, and this is still under testing. We are looking for Parkinson patient subject to volunteers. So we are doing the clinical trial these days, so we can arrange you to be one of the subjects of this sweat monitoring. So this going on. So please contact Professor Irene Litvan or contact me. I can connect you and we will do the testing, either the sweat or the future will be the microneedle. Ultimately, it will be a closed loop, as I mentioned, with microneedle, with Dopa pump that you can deliver autonomously on demand. Yeah.
Dr. Wang, can you repeat your email one more time?
It's in my name, Joseph Wang, lowercase, at ucsd.edu. Joseph Wang at ucsd.edu.
Perfect. Thank you. Another question is there any tie-in into monitoring other cancers or adjusting chemo?
Yeah, eventually we are moving. This is what I gave you flavor of few examples. There are a lot of activity of different disease, cardiac disease. Now, in the case of cancer biomarkers, they are not changing so fast. So it's not like cardiac biomarker, you need the quick action. So we are looking for troponin or kidney biomarker, creatinine. The cancer biomarker are slowly moving. So it's not something quickly you see the up and down change. So you can do testing in the lab without going continuously, like in the case of cardiac or Dopa or diabetic. But we are having sensor we are developing for surgery, where we are looking for the anesthesia, the propofol, and the painkiller. So we are working on, for cancer treatment, the surgeon wants us to develop sensor that during the surgery they monitor the anesthesia level, the drug propofol or the painkiller, because you have a very narrow therapeutic range and need to adjust it very carefully.
Thank you. There's a question about sharing your PowerPoint presentation. However, we will be posting this recording on our website, which then you can see again and see Dr. Wang's presentation on. Okay.
And again, feel free if you have any question or any interest to collaborate, invest, please, we'll be happy to join force and discuss potential collaboration and clarify more about the technology. A lot of this is on my website, and you Google Joseph Wang UCSD, you see on the research, a lot of examples, but specific Parkinson diabetic, I can send you and we can discuss potential collaboration.
Great, thank you. Dr. Wang, how soon do you think this technology will be available to the medical community?
As I mentioned, a company like BioLink here in La Jolla, they're very close. They do the final FDA clinical trials. Hopefully, by the end of 2023, in 16, 18 months, they will launch their first microneedle-based, what they call CGM. Again, I told you about the Swedish companies that do it for wellness, also diabetic and wellness in Sweden. This is Innovosense. There is Australian companies that do for Nutromic, for the monitor nutrition and drugs. So we are pushing the more startup company from my lab. Everything takes time, especially the medical need further FDA approval. But for wellness and sport nutrition, it's getting closer, less regulation. In general, all these non-invasive make it simpler to get the approval compared to anything invasive. Even microneedle still slight penetrating the skin. If you can do it continuously, non-invasive, you don't have this issue of the regulation.
Dr. Wang, do you think that this sweat analysis on the skin will be the first device we see on the market?
No, I think the microneedle are getting closer. So because there have been activity like BioLink started the journey 8 years ago already. So they are pushing. But we predict sweat will be also very soon. And the Swedish company is for sweat. This is Novosense. So they just launched the product. Yeah.
Great. Well, thank you, Dr. Wang. That was our last question. Thank you so much for being part of our distinguished speaker series. If anyone has any additional questions for Dr. Wang, you can email him at josephwang@ucsd.edu. You can also access a recording of this presentation on our website, which again is ljcommunitycenter.org. You can find out more information about our center and our programs that we offer there daily. So thank you again all for joining us.
Thank you, Nancy. Thank you, everyone. Thank you for joining, and we'll keep in touch. Enjoy. Beautiful day in La Jolla. Bye-bye.
Absolutely. Have a great day.